Are you taking AP Biology? If you are, you're in the right place. I'm here with Tiffany Jones. Oh, here.
Hi.
I think you're here. From
I'm
AP.
there.
I don't know where you are. Wherever you are, you're amazing. And I'm really excited to be hosting you here. Everyone, if you don't follow AP Bio Penguins on Instagram, what's wrong with you? Go there now. I'm going to show you, I'm going to share my screen. This is the place to be when you want to study for the AP Biology exam. Tiffany, tell us, what are some of the really awesome things that you have on this channel for everyone?
So starting in February, I do a daily stories quiz, like where I put multiple choice and short answer questions in there. Every Friday, I go live for FRQ Fridays and go through 2 or more free response questions. And then twice a month, I go live with the Absolute Recap to do a Q&A session. We actually have one starting tomorrow on Sunday at 8 p.m.
Amazing. So there's questions, quizzes, there's links in the link tree to some of the incredible resources that you have about AP exam review materials, the schedule of what you're doing. I'm super excited to have you here today. We're going to be going over Unit 3 of AP Biology. Now, when I was in high school, I took AP Biology. True story. I got a 4 in the exam, which I'm very proud of, because I didn't get a 5. One of the reasons why was that I hate plants. And while I was in AP Biology, I told Mrs. Potts, my AP Biology teacher, I said, I love you and I love this book, but I don't like the chapters on plants. So I'm going to try to get the highest score I can get without, even though I'm like plantophobic, and I just refused to participate. So I remember a couple free response questions where I was like, I don't know, stamen, pistil, leaf, whatever. What is Unit 3 all about? That was a different curriculum. It was a different AP Bio exam. What are we going to be discussing about Unit 3?
Unit 3 is about enzymatic activities, cellular respiration, and photosynthesis, which students tend to find that cellular respiration and photosynthesis are their hardest units. So I want to make sure that we are going to get the students as much as we can to help them with what they feel is their hardest unit.
Absolutely. That's awesome. So, and I see the Absolute Recap is in the chat with us live. So definitely check out the Absolute Recap. I'm actually going to pull up their page real quick on Instagram. If you don't follow them, and let me just see.
There's a podcast, and they do podcasts for AP Biology, AP Chemistry, Physics 1, Government, and then Music Theory. And so it's one for each day of the week, and they release them on the same day. I can't remember. I know Biology is Wednesdays.
Yeah,
and then she posts the question of the day.
Yeah, I'm sorry, I'm interrupting. What I love about this, I'm very passionate about this, because these podcasts, one, are awesome. They've got amazing AP teachers like you, Tiffany, and podcasts are free. So you work through the podcast, and then there's these study guides that you can download from it. So definitely check out the Absolute Recap. You put the at the front, AP Salute Recap, and follow them on Instagram and on the channels where they're putting out their podcast. Podcasts are great, everyone, because we're all spending too much time on screens. So put those AirPods or buds or whatever in, and you can do something for 10 minutes and kind of really be in that moment. So Tiffany, I'm going to turn it over to you for Unit 3. Let's get right into it. What are some of the exam hacks that can help me not get a 4 this time, but get a 5?
Help you get a 5. Well, first thing, before I go into it, I want to tell you why my students are penguins and why every single student who's currently watching this is a penguin. Well, penguins are, of course, dressed for success. So as an AP Bio penguin, you are dressed for success. So now that you are watching this video, you are an AP Bio penguin. He said he wanted me to go over just my favorite hacks. Okay. So he already mentioned about the AP Bio penguins. I have stuff on Instagram. That's where my main platform is. But I also have been putting TikTok videos together and popping them onto Twitter, and then also on YouTube. I put all of the live streams that I do with the Absolute Recap, and all of my Friday FRQs are all on YouTube. I have a website, apbiopenguins.weebly.com. And on there's a ton of resources. I'm throwing up all of the, I'm putting up all of the review PowerPoints from previous years. I'm putting up the a 245-page review guide that I made that breaks down every single unit for you. Other resources that I find to be helpful, Absolute Recap, as we already mentioned, is a podcast. Stick it in your ear and go ahead and go for a walk. You can walk Marco if you want, John. Also, my favorite YouTuber would be Bozeman Biology. Paul Anderson, genius. And so he has a lot of really good videos that really get to the AP level, unlike some of the other videos that are out there. And then if you really wanted a review book, Barron's 7th edition is the most up-to-date version that we have in terms of review book. So you could read the sections that you have trouble with and then do the little quizzes at the end and kind of check your knowledge. So before we get into cell respiration and photosynthesis, let's kind of talk about the exam, right? There's 2 different exams that we're going to possibly have. You can either have the paper administration or the digital administration. My school is offering both of them. You can either take paper or digital depending on your own needs, your own wants. My school is mostly virtual, so they get to make the decision. So I've heard that depending on your school, you might not get that opportunity, students. But so if you're taking the paper administration, which is what I recommend, because you can freely move throughout your multiple choice section with no issues. You can also freely move around your free response section. You can manipulate the paper. You can circle things. You can underline things. You can make marks on it. And that will be on May 14th. And then if you're taking the digital exam, that will be on the 27th or June 11th. Both are going to be at 12 o'clock Eastern time. No matter where you are in the world, they're all going to be taking it at the same time as part of the security that they're putting on the exam. It's the same length in which there's 60 multiple choice questions, and then we have our 2 long and 4 short responses. But instead of actually graphing anything or having to draw anything, it'll already be embedded in the question somehow. So it's the exact same exam, just there are slight differences. The big thing of why I believe the digital won't be as successful or successful for you, or is going to be a harder exam for you, is because you cannot move on. Once you move past a question, you're stuck. You cannot go back. So if you're on number 4 and you realize that you made an error on number 1, you can't go back to number 1 to fix that. And if your brain is anything like my brain, if I don't know the answer, I move on. And then later on, my brain kind of thinks about it, and then I can jump back to that question and answer it. That will not be able to be done during the digital exam. Same thing as you can't manipulate the paper, you can't circle things, you can't underline things, which to me is a disadvantage. So some quick tips for Marco. Is that number 1, pace yourself. We have 10 weeks until the AP exam. If you were to take the first paper exam, it's on Friday. And so basically 9 weeks and 6 days is when your exam is. There are 8 units. You can do 1 unit a week between now and the AP exam, and there's still time for you to do a practice exam in there and do some practice problems and kind of really slowly review. I find that a little bit a day is the best way to review. You want to be present practice. What that means is that when you're doing your practice problems, you want to be in an environment similar to what your testing environment is going to be. You want to be quiet in your room. You want to be, working on a piece of paper. If you're going to do a paper exam, you want to be working on the internet. If you're doing a digital exam, you want to model what you're going to be under in those conditions, especially when you're doing those practice questions. Also, present practice means that if you are doing a problem or you're looking over the answers, you should go back and figure out why you got things wrong, okay? Don't just take it and go, oh, I got a 50 on this practice, moving on. That doesn't help you. Go back to the questions you get wrong and go and figure out, well, why is this wrong? Why was this question not correct? Especially if you're using one of the Barron's books, they've got explanations in the back of the book. It'll explain why that's the answer, okay? It's a perfect way to review. Persevere. Biology is hard. Okay, I'm not going to be the one that tells you that it's not, because it is. And John got a 4 on the exam. Great job, John. But you need to persevere. Push yourself through it. Okay. If I remember doing the math for our jumpstart session, and I think that it was like a 54 out of 120 points that you need to get on the exam in order to make a 3, and that was based on the 2020 practice exams that we had, which is less than a 50%. So don't be upset if you get through a practice and you get a 60 on it. You're looking pretty. You're doing a great job. You want to, of course, be as successful as you can be, get as high as you can get, as many points as you can get, but persevere, push yourself through it. Okay. You don't have to be perfect. You don't have to make 100% on every single task to be successful. And last but not least, you're a penguin. You're dressed for success. You've got this. I believe in you. You have to believe in yourself. So As I said, today we're going to look at Unit 3. I'm not going to do a lot of enzymes, but I did put that on there just so that we knew that enzymes is within this unit. So we're going to focus on cellular respiration and photosynthesis. And Marco wants to remind us that 12 to 18% of the exam is this unit. And so when I went through the 2020 practice exams, there were 3 of them released to teachers. If your teacher has not given you one, or your teacher hasn't planned for there to be one in the future, just give them a reminder. Say, hey, I'd like to be able to practice exam before I take it. Is there a Saturday that we can meet for us to do this? Is there that I can stay after school and do it? Can you assign it to me in some way? We have access to during a online class. We can put it through AP Classroom with the lockdown browser. So there is a way for you to take a practice exam. So be sure that your teacher does allow you to get one of those practice exams. But based on those 3 practice exams, there is 9.3 questions that was in this unit. I went through and actually analyzed the test to see what units everything was from. So let's dive into it. Cell respiration is heavy, right? There's 10 steps to glycolysis. There's 8 steps in the Krebs cycle. There are so many little pieces. There's all these enzymes. There's all the intermediates. That's a lot to look at. Let me ease your mind a little bit. Cellular respiration, you need the big picture. What goes into the reaction? What comes out of the reaction? Where does it take place? And why is it important? Okay, that's what you should know about each of the steps. Do not worry about every single enzyme. Do not worry about every single intermediate. You need the overall understanding for this question, or these questions you'll see. Okay, so when we think about the first step of cellular respiration, which is glycolysis, and if you use your terminology, you can kind of break this apart. You've seen lysis before, right? You saw hydrolysis, where you're going to use water and break a bond. You saw photolysis, photosynthesis, where you're going to break a water molecule up in order for photosynthesis, okay? You saw lysis of the cell when we talked about diffusion and osmosis and how the cell is going to burst open, okay? So glycolysis is our sugar splitting step. We will start with glucose. It is a 6-carbon structure, okay? And the output, what comes out of it is, I always tell my students, is a 2-2-2. You get 2 NADHs, 2 ATPs, and 2 pyruvates. You had to put in 2 ATPs and you got technically out 4 ATPs, but like it's a net gain of 2. So we just focus on that it's 2 NADHs. And the lie that I tell my students is that H stands for holding. And so what that NADH is doing is it's holding those high energy electrons that were in glucose. And as we break down that glucose, we absorb those electrons and we hold them. That's what the H is holding the electrons. And then we're going to transport that over to another step in which we can use that high energy to fuel a reaction. The ATP is made by substrate level phosphorylation. What that means is that at the substrate level, there is a molecule and that binds to an enzyme. So at that point, we can add the phosphate to ADP. When we say that we're synthesizing ATP, all we're doing is adding a phosphate to an ADP. Okay. And in case you don't know what that stands for, ADP is adenosine diphosphate, and then ATP is adenosine triphosphate. The only difference, we added a phosphate to it, which is how you got from ADP into ATP. And that's occurring at the substrate level. And then we have our pyruvate, which is a 3-carbon molecule. And so if you need help kind of remembering this, we had started with 6 carbons, right, in glucose. And then through this process, we now have 2 3-carbon structures. We did not lose any of our carbons. So you know that there's no carbon dioxide that's been lost at this point because of the fact that I still have all 6 of my carbons. They're in 2 different structures, but I still have all 6 of them. Where does this take place? It happens in the cytosol. And this was a question somebody asked me before, is what's the difference between the cytosol and the cytoplasm? Big difference? Cytoplasm is organelles, cytosol doesn't. Okay, it's the same exact location. So you can probably use these 2 words interchangeably with no problem. But I always think of it like a fruit cup. So within your fruit cup, you have all that fruit in there, right? That would be your organelles. And then like the watery substance that's in the fruit cup, that's your cytosol. So together the fruit cup is cytoplasm, but just the liquid would be your cytosol. And so why is glycolysis important? Why do we need to know about glycolysis? Well, number one, glycolysis starts this process of breaking down the glucose so that we can get that energy out of it, okay? And so if step 3 that I have over there is breaks down the glucose to pyruvate, and also allows us to get those electrons, the NADH, and that carries it over to the electron transport chain, which is what ETC stands for. And last but not least is important because I made ATP. So organisms that can't go through the other 2 steps of cellular respiration are able to make ATP. They're able to make those 2 molecules of ATP, and then they go through a process called fermentation to regenerate the NAD plus, which is going to be the oxidized form of NADH. So it goes between NAD plus and NADH. And then also important about this is the fact that since we're talking about it being in the cytosol, all organisms can go through this process, and you can hear my son in the background. I'm so sorry. So we have this intermediate step, and I usually don't talk about the intermediate step because of the fact that it's kind of a component of glycolysis. You kind of think about them literally being attached. It's like literally just an intermediate step. But this past week when I was going over it on the Instagram, students were freaking out. So I wanted to go ahead and throw this up there as like, put this up there as another kind of slide so that you could make sure that you know about this step because students were a little antsy about it. So the input for the intermediate step, which is called pyruvate oxidation. Some people call it the link reaction. Some people just call it intermediate. Some people call it the grooming phase. It doesn't matter what you call it as long as you know what happens in it. So pyruvate oxidation, we're going to start with pyruvate, which was the product from glycolysis. And we're going to get another electron from it. And so we'll have 1 NADH. And then we're going to break off a carbon dioxide. So the carbon will come off. So now I have a 2-carbon molecule. And if you know your prefixes, acetyl stands for basically 2 carbons. And then that CoA is a coenzyme. And so they added a coenzyme to it to kind of keep it in this higher energy state so they can go into the Krebs cycle to continue the process. Where does it happen? Mitochondrial matrix. So if you don't have the capability to bring this pyruvate into your mitochondria, you're not going to be able to break it down. So I've seen questions in which they talk about how does the pyruvate get in? Well, there has to be a membrane protein. There has to be a channel protein because pyruvate is polar. And so in order to get it through a membrane, it has to have some type of transport protein to get it in. And so if a organism doesn't have that transport protein, they could connect it back to, well, this organism must go through fermentation to regenerate the NAD+, and they only go through glycolysis. They'll have a lower amount of ATP. And you can talk about that the organism might be lethargic and have a higher level of lactic acid, and there would be all this other byproduct since it couldn't get into the mitochondria. And why is this step important? It makes the NADH carry that to the electron transport chain. It also prepares the pyruvate for our Krebs cycle. So Krebs cycle, also known as citric acid cycle, also known as the tricarboxylic acid cycle. So I've seen that students sometimes have trouble remembering. And so it is important that you know that these 2 are the exact same thing, or actually all 3 of them. I've never seen the tricarboxylic acid cycle on the exam, but you do need to know that Krebs cycle and citric acid cycle are the exact same cycle. There was an exam question, which we're going to do in a little while, in which at the reading, teachers were telling the item development committee, like, my students didn't know that those were the same. Well, that is your issue, not anyone else's issue, okay? You're being the teacher's issue, not you, the student, okay? You need to be aware that these are actually the same thing, okay? In this cycle, Krebs cycle is going to have an input of acetyl-CoA, which came from that pyruvate oxidation. It is made up of 2 carbons. And in this process, we will completely break down that acetyl-CoA. So we will end up with 2 carbon dioxides from every single acetyl-CoA. The numbers I have on here are based on 1 acetyl-CoA. But we know that in glycolysis, we made 2 pyruvates, which means technically in the full breakdown of glucose, there's actually 2 acetyl-CoAs. And so you would actually double all of this in terms of the question being for glucose, not acetyl-CoA. But I just wanted to mention just based on what we have, which would be 1 acetyl-CoA, gives you 3 NADHs, 2 carbon dioxides, 1 FADH2, and 1 ATP. And you're like, wait a minute, where did this new word FADH2 come from? FADH2 is just like NADH. It's still holding the electrons. It's just not as strong. So it kind of ends up lower on the electron transport chain, and it will generate less ATPs. This happens in the mitochondrial matrix. And then the reasons why it's important, it makes the NADH and FADH2 in order to bring it over to the electron transport chain. It completes that breakdown of the glucose. And also we see that substrate level phosphorylation to make ATP, which is actually a lie because we make GTP, which is the exact same, except for instead of having an adenine, it has a guanine. So we just kind of simplify it by saying, well, there's an ATP. So I hope that this is making okay. John's got the chat, so if you're writing stuff in the chat, I can't see it at this moment, but John will cut in a second and tell me what's going on. So our last step of cellular respiration is oxidative phosphorylation, which has two different parts to it. You have the electron transport chain, and you have chemiosmosis. The electron transport chain is exactly what it says. It is an electron transported down a chain, and as it moves down this chain, it's going to pump protons across the membrane. Okay, so the NADH comes in as this high energy molecule. Okay, and as it goes through the process and it changes hands and goes through the redox reactions of each one alternating, the electron is going to slowly drop in energy. Where does that energy go? The energy goes to pumping a proton against its concentration gradient. So it's being stored as a potential energy on one side of the membrane. And so that's what the whole thing about electron transport chain is. At the very end, that oxygen molecule is going to bind to the electron and it will make H2O. Now here's the thing. At this point, we have not made any ATP. ATP was not made in the electron transport chain. There was no ATP made in the electron transport chain. The ETC, electron transport chain, its sole purpose is to make that proton gradient. In chemiosmosis, the second part of oxidative phosphorylation, is where that proton gradient is going to allow the protons to flow down their concentration gradient through ATP synthase. It's an enzyme. Remember, enzymes tell you what they do, right? ATP synthase is going to synthesize ATP. It's going to take that energy that's released from the proton coming down the concentration gradient, and that is going to fuel the addition of a phosphate to ADP to make ATP. And this is where most of the ATP is made in the process of cellular respiration. And so where does it happen? Mitochondrial cristae, which is that inner membrane that you have. So if you remember your mitochondria, there's like that outer membrane and there's like the squiggly thing on the inside. That squiggly thing on the inside, that is your cristae or your inner membrane. Why is it important? This is where we get the electrons in order to provide the proton pumps to get that proton gradient. And then this is also where we get the highest amount of ATP. So looking at that membrane is a super important concept, okay? And it's important because it connects to other things. So as I said, electron transport chain does not make AT. The electron transport chain's role is to produce that proton gradient. Okay, so as you see here, and I hope my mouse is showing, here's our electron transport chain. The NADH drops off its electron and it's going to go down the concentration, kind of going down in energy. And as it does it, it pumps these protons across. Notice there's a lower concentration here and a higher concentration here. We're pumping it. Now it's important to know which direction it pumps. It pumps it out into the intermembrane space. So the spaces between the two, so we have that outer membrane of the mitochondria and that little inner part, it's in that between it. It's the intermembrane. It's a space between two membranes. And so as it does that, it increases the concentration of the protons, which decreases the pH. So you could have questions about pH and the mitochondria because we're looking at these protons increasing in concentration. And if you understand the way pH works, you know that means that the pH is decreasing. So that's an important thing to note. And also here you can see those protons come down their concentration gradient through this enzyme called ATP synthase, which adds that third phosphate. So Mark, I'm sorry, John, I put a little pause here so that we could kind of let our brains kind of have a moment. And I think John's there. Hi, John.
I am. Yeah, and Tiffany, this is so great. I've got a couple questions from the chat and some ideas I wanted to follow up on, but this is a very important slide. On our Instagram page on Marco Learning, I put up a reel asking for what great AP teachers are out there. And we were bombarded with hundreds. Apparently there are thousands of great AP teachers in America. And all these people, Tiffany, these people were marked as AP biology teachers, right? Mm-hmm. So we have Mrs. Fowler, Mr. Heinrichs, Mr. Hansen, Mr. Davis, Mr. Huff, Mrs. Erdman, Miss Mina, Ms. Jewett, Dr. Long, Ms. Rivera, Mrs. Kimbrell, Mrs. Sherman, Mrs. Sharp, and Mrs. Jones. Are you Mrs. Jones? Did you put your...
It wasn't even. No, somebody did put my name on there, and it wasn't even, and it upset me. My little type A pop like style was like, oh, we need to have them even. So I was like, I'll then put my name there. But my name originally was not up there, but I swear it's on there.
No, I know it is. Well, look, on Instagram, Tiffany, you're doing incredible work on the AP Bio Penguins page. When I was live for AP World History the other day, remember, we're going live on this channel every single day in the month of March. I was live with Emily Glankler from Antisocial Studies, and she was convinced that her sister had put her name on there, but it was in fact one of her students. So guys, I think it's a great time to celebrate AP teachers. I teach 6th, 9th, and 12th grade. It's a tough year this year. It's really hard to do our jobs with everything going on. So we really want to thank all of the teachers out there, everyone at Absolute Recap, everyone at AP Bio Penguins, everyone at Marco Learning who is trying to help, and all these wonderful full teachers here. So definitely add more comments and names, and we will shout those teachers out. Tiffany, we've got a question in the chat from, and I'm going to just take this down for just a minute. We've got a question in the chat from Ria, which is, why does fermentation occur when we do intense exercise? Is it because it is a lot faster? So that's an interesting question. One, well, not for me because I don't exercise, but I'm curious, maybe this is the reason I don't exercise, right? Lactic acid fermentation, is that what happens? So yeah. Why does that occur? Is it because of the speed of what we're doing?
It's because of the fact that in order for the muscle to contract, it requires ATP. And so the ATP has to be a quick process. And so it has to do with the trypsin and the tryptomyosin and its binding and all that fun stuff. Anyways, but so when you go through exercise, you do it so fast that there's not enough oxygen in your muscles. And there is this thing called myoglobin. So we know about hemoglobin and how that brings oxygen to the cell, to the whole body. But myoglobin is a specific oxygen holder that occurs in your muscle cells. The myo at the beginning tells you muscle. And so that usually is a storage of oxygen. But if you work out too long, you use up that storage and there's not enough oxygen getting to your muscles, which causes your cells to go through lactic acid fermentation, which is why your muscles burn. Like if you've ever stopped, once you stop working out and they like burn and they hurt, you're like, oh, and they always tell you, you should go work out again. And you're like, no, it hurts. It hurts because of lactic acid. And the only way to get rid of it, or the fastest way to get rid of it, is to work out and get the blood pumping through the area to move the lactic acid to your liver to convert it back out.
All that, so, yeah. That sounds very exhausting. It's a validation that I don't exercise and take care of myself at all. So no, this is really interesting because I think what we're seeing here is that this stuff that you guys are all going to be studying over the next few weeks for AP Biology is relevant to our everyday lives. It is literally in our bodies right now. And so a couple other things real quick. For everyone who's joining, if you like this video, press that like button, subscribe to our channel. We are going live. Tiffany's going to be live on AP Bio Penguins on Instagram. We're going live together on this channel several more times in the month of March. We'll be posting those announcements on Instagram and on the community page. And we got a great question from Ria. We've had some hellos in the chat, but definitely post your questions. Tiffany and I are here to answer them. 2021 is a strange year. We have the digital and paper and pencil exams. There's lots of complexity. Remember, on our channel, we have videos walking you through those changes, walking you through the digital exam. So Tiffany, I'm gonna let you share and kind of walk us through the next hack here for Unit 3. And it looks like we've got plenty of time to cover everything. So I'm gonna turn it over to you and we'll keep going.
Hey. So the plan, where's the call machine? The plan I had was, let's go ahead and immediately apply this. Like, let's go ahead and think through an actual practice question on cellular respiration. So it says, according to the data, the mice at 10 degrees Celsius demonstrated greater oxygen consumption per gram of tissue than did the mice at 25 degrees Celsius. It's most likely explained by which of the following statements. And then it gives you a bunch of statements, okay? So my advice for students is, before you look at answer choices, go ahead and come up with an answer yourself, right? Because I find for myself that when I try to do a problem, the answers all sound right. It's like, I look at them like, yeah, that one's right. Oh, wait, no, that one's right. Oh, that one's right. And I convince myself what the answer is based on which one sounds the best. And You know your biology, you know your information. So go ahead and formulate what you think the answer is before you read the answer choices, okay? And that will allow you that if you see your answer in the answer choices, there's a higher chance that that's the correct answer. And so we know 10 degrees is is a lower temperature than 25 degrees Celsius. We know that mice are something called endotherms. What that means is that they're warm-blooded organisms. Your middle school teacher calls them warm-blooded. Your high schooler is like, what is that word? That just means that these organisms generate heat through their metabolism. So if it's a colder temperature, you're going to have to go through more metabolism in order to generate enough heat to keep your body temperature constant. Endotherms go through the metabolism to get that body warmth, versus something called an ectotherm, like a lizard, is going to move into a warmer area to warm their body up. That's why I think it was last year, the year before, we saw a whole bunch of lizards and iguanas falling out of trees in Florida because it got too cold, and they pretty much just kind of like semi-froze. Like they weren't frozen, but they were like slowly moving. And so we know that if I have a higher amount of metabolism, 10 degrees Celsius than 25 degrees, that means that I'm going to go through the process of cellular respiration more frequently. Well, oxygen was a final electron acceptor. If I go through more metabolism, I should theoretically use more oxygen, right? And so the answer choice A, thanks Marco, is that the mice at 10 degrees had a higher rate of ATP production than mice at 25 degrees because they're going through cellular respiration more frequently. And don't you like my little Marco over there? He gave me the answer. Super cute. I don't know. I was having a little too much fun this morning at 3 a.m. So I said before that you didn't really need to know all of the major information about cellular respiration, right? You had to get an overview. Well, here is a free response question from 2013. I don't remember the year, sorry. And they gave you this diagram. This diagram was copied and pasted from the exam, okay? And so you're told by looking at this, I can see, okay, look, there's glucose, I can see this pyruvate, I can see the acetyl-CoA, I can see, this whole cycle here. I can see what's going in, I can see what's going out, I can see this process here. Like, so they gave it to you. They gave you a visual. Use your diagrams to your advantage, okay? See what you can pull from the diagram, especially if you don't know the answer, okay? So the question was, using the information, describe one contribution of each in ATP synthesis. So if we look at the first step, it says catabolism of glucose and glycolysis and pyruvate oxidation. So looking here, what happened to make ATP? Oh, look, ATP is in the picture. This process produces ATP. So you had to bring in a little bit of biology by substrate level phosphorylation. Oh wait, but I don't know anything about that. Okay, well here's the NADH, and I see, look, down here, there's NADH. NADH was formed in order to go over to electron transport chain to then make ATP. You can use your diagrams to your advantage. Oxidation of intermediates in the Krebs cycle. So here we see this Krebs cycle, and again, I made GTP, which we said was the same as ATP. So again, substrate level phosphorylation. You also see NADH. It goes to electron transport chain. So use your diagrams to your advantage. And last but not least, that formation of the proton gradient by electron transport chain. In this process, you can see that there's a proton gradient that's been formed. That proton gradient is going to fuel the process of ATP synthesis, okay? And so this was the scoring guidelines. You could have given any number of those answers, talking about the ATP produced by substrate level phosphorylation, talking about the NADH and the FADH2, talked about the GTP by substrate level phosphorylation, talking about how allowing those protons to flow down their concentration gradient and how they had this potential energy. And so based on what we talked about earlier, you should have been pro at this step, right, on A. And you can screenshot if you need to, but it's, I think the 2013 number 1, maybe 2002, I don't know. So the next part, this is using each of the following observations to justify the claim that glycolysis was first occurred in common ancestors. So wait a minute, I thought we were on cellular respiration. How did we jump into evolution? The questions you're going to see on the AP exam, they don't just stay in one unit. They jump between multiple units. They encompass the entire year. So yeah, this is an evolution question, but it also has to do with cellular respiration. And so it kind of is married in this question, right? Nearly all organisms perform glycolysis. We said that before. Well, how do I know that? Glycolysis occurs in the cytosol, which would have happened before the, in kind of the prehistoric cells, before a mitochondria was made. So that allows us to say, okay, well, it occurred in cytosol. All organisms go through the cytosol. This has to have happened before membrane-bound organelles. Glycolysis is an anaerobic condition. did you see oxygen used in glycolysis? Well, there's no oxygen used there, which means that it occurred before oxygen was present in the atmosphere. And last but not least, it occurs only in the cytosol. Wait, I must have jumped around something. Oh, I already said that part. That was me talking about the cytosol. The first one was supposed to be about that all organisms perform glycolysis. The cells that were able to go through glycolysis could produce more ATP, and that trait must have been passed down. And so, of course, and I kind of stretched this out just to make sure it was big enough. When we were doing our AP jump starts, John reminded me, like, okay, make sure that it's big enough for them to see. So I tried to make the pictures as big as I could, which kind of made the fonts a little funky. But talk about how it is passed down from generation, talk about how there's a selective advantage. Those individuals who are able to go through glycolysis could make that ATP versus other ones had no energy source or they had to go through some other way. Talking about that it predates the atmospheric oxygen, also saying that it predates any type of membrane bound organelle. So just kind of applying the concepts. And then you're going to have math. And I know that you don't want to do math, but sorry, biology does a lot of things. And one of the things we do is math. The quick view of this thing is saying that if you break down the glucose, you end up with 30 molecules of ATP. If you were to just break down glucose in the lab, you should have 686 kilocalories per mole of glucose. And then each ATP is actually 7.3 kilocalories per mole. So you're supposed to calculate the energy that's available from hydrolysizing 30 moles of ATP and calculate the efficiency of that ATP production from 1 mole of glucose, and then describe what happens to the excess energy. Like, whoa, what do you want me to do? Well, so we know that we have 30 molecules of ATP, right? And each one is 7.3 kilocalories per mole. So all you have to do is say 30 times 7.3, and that tells you that there was 219 kilocalories from like stored within your ATP, right? And you can use and do something with it, right? Well, that doesn't make any sense because glucose has 686. So what's the efficiency? Well, all I just got to do is divide it, right? I had 219 kilocalories that I was able to use. And then there was a total of 686. So just make the ratio. It gives you 0.319. And both of these answers were accepted. You could have said 31% or 32%, depending on how you round. Students have trouble with rounding. FYI, this is a 9, so it should round up. But they do take into account that you might have slight rounding. So sometimes they'll allow a range of 31 or 32. They'll round to kind of an up or down. And all the extra energy we mentioned before with the endotherms, excess energy is released as heat. That's how you generate your energy within your body. And this is also how all of the other systems, think about your car, right? You go and you drive your car, and then when you get to wherever you're going, right? If you touch the engine, is that engine cold? Nope. That engine is nice and warm. Why? Because that excess energy from the combustion of your gasoline is producing heat, which heats up your engine, which is why you have like the coolant and those radiator. I don't know anything about cars. I'm not even gonna try. So yeah, all this talk about solar respiration. Marco, you want a snack? John, you got Marco around so you have like a snack break?
Yes, let's do some puppy ASMR right now. I'm gonna go get Marco. Well, he's kind of sleeping right over here. Marco.
Oh no.
Marco. Let's get him up here. And I'll take this down for a second.
And I know that you probably don't want to give him any more treats because last time I watched the video with Emily and you said how heavy he's getting from all these snacks.
He is getting he needs to do some more cellular respiration or something. A little bit more lactic acid fermentation. Here, I'll bring him up here.
And I hope that this has been helpful for everyone. Oh my gosh, Marco, he's so fluffy.
Now, let's get him up on here. Marco, no, please. He's trying to eat it off the desk. Marco, the people want to see you eat this. Come here. Your fans want to see you. There we go. There we go. That's a little.
Good job, Marco. You've done so good today.
Treat. Isn't he a good boy? So we are getting some interesting discussions. I would hoist him up more, but he will. There he is. You see, he is quite fluffy, and he has his little paws ready to type for his digital AP exam. I'm going to put him down here. One of the things we're seeing in the chat, first of all, I hope you all got your snack break. Yes, and some people, Ria's talking about getting her chocolate now. Ria also asked a follow-up about the exercise question, just stepping back to that for a second, that drinking water, which I'm sure Marco wants to do after his little lamb treat, reduces the power of enzymes. Does it have something to do with water and diluting reactant concentration? Does drinking water while eating, reduce the power of enzymes? Or is that something we've got to follow up with? Look up.
I don't know the answer to that question. I might have to follow that up, but I'm going to go with a biological background and see if maybe I can guesstimate an answer. Yeah. Water, although is going to dilute your acids, which are parting for the digestion, water is also needed in order for digestion because we go through hydrolysis, which breaks the water molecule. And by breaking the water molecule, you can attach the hydrogen to one side and the hydroxyl to the other. So I don't know why taking more water would have issues with digestion. I will write that down and I will have it posted on my page.
Tiffany, what I think is so interesting about what you just did is you acted like a really smart penguin, a really smart AP Bio student, because true story about AP Biology, everyone, you don't need to get 100% of the questions right to get a 5. You don't need to get, 75% of the questions right to put you in that range. And so this is one of the only standardized tests in the world, AP Bio and the whole AP system, that will give you a perfect score for something like a C, right? On the AP histories, if you're 74% on the multiple choice and 70-ish% of the points, you're gonna get a 5. That's astonishing. And so it means that
that's a little higher, but yeah, I think we're about 80%.
80. So I think
about 80.
If I want to get a 5 in AP Bio, I need to get a B-, at least this school, right? So I need a B-, and I need to get around 80% of those points on the thing. But that's something that you're taking practice and you get 67% correct, and you're like, I literally failed this section. You're very close, and I think with weeks of studying, you can develop a plan to start going after those areas of weakness. And then what we just saw Tiffany do, which is they'll throw you kind of a curveball, a question you weren't expecting, and you kind of reason it out, write out that work, get what you need done, and try to go for the points that you can get, rather than feel like, I got to know everything. Tiffany, for that digital exam, it's going to be interesting because you're clicking through and you have to be decisive because you can't go back.
Right. And what you were just mentioning, that's actually how I write my free responses. The question they're going to give you is something completely off the wall. You will have never seen this. It's going to be something completely your teacher can't prepare you for it. It's going to be all applications. And so when I'm trying to justify or reason through something, I actually will write it all as part of my answer choice. And then I accidentally can fall into points. And as a reader, I see students do this all the time. You're just trying to logically write it out, and then all of a sudden you just happen to get a point. Points game, right? Wherever you get the points, you get the points. It doesn't matter how, just get those points.
Exactly. Like Ria's saying in the chat, the key is to figure out the test. And that's where really intentional practice, really studying yourself as a test taker, and crucially doing what you're doing right now, which is hopefully, maybe some of you are watching the recording the night before the test, but starting early with a plan. So Tiffany, I'm going to invite you to take us through that third segment. We're going to wrap up here within the next 5 or 6 minutes, but just some final thoughts and hacks. And remember, we're going to be going live again on this channel, and Tiffany is going live on her AP Bio Penguins.
So do you want me to skip photosynthesis and go straight to my hack?
I think we might need to. We'll need to wrap up in the next 5 or 6 minutes here. So
we'll skip all that.
Great. We could cover some of this in our next review as well.
Okay. So strategies for your questions then, because we're wrapping up in the next 5 minutes or so. Use your diagrams, as I said before. Okay. A lot of times these diagrams will give you the answer. So on the first response, the first response that we didn't actually get to because we jumped ahead, it had to do with you had to identify which one had the highest rate of photosynthesis. Well, make a guess, right? If you don't know, just guess. It says to identify, you look at the diagram, you can pick 3 things out, oh, this one's highest, medium, low. Maybe you get that point. Maybe you don't. It's okay. You gotta do your best, right? So develop your own ideas. So as you're going through these questions, come up with what you believe the answers are. When you're looking at your graphs, go ahead and say, oh, I see that there's this increase. As the concentration of enzyme increases, the reaction rate increases. And you can kind of see these trends in your graphs. If you notice some weird number in a table, circle that number and kind of make yourself a note and be like, what is going on here? Like question mark, question mark, question mark. When you see that there is this material that's working in the glycolytic, you say, oh, well, this is, you know, cytosolic protein. Like it kind of make notes on your diagrams. Okay. So my paper testers, you want to underline and circle any important information in the prompt. You can manipulate your test. You can work with it. So just do whatever you can on that paper to try to make sure that you don't have to reread that prompt. You only have 90 minutes for either of these sections. So you want to make sure that you don't have to reread these prompts. And these prompts are sometimes dense. There's a lot of biology in there because there's no required labs, right? There's no way for me to sit here and say, well, the exam question is going to have this lab, this lab, this lab on it. You don't know which one it's going to be, and they have to give you the background information so you can apply it. If you've done a lab, awesome. If you haven't, they had to give you enough information. So those prompts are kind of dense as they're giving you all this information you're going to need. So underline, circle, whatever you can. Write your analysis for your graphs on your graph so that you can kind of see what's going on. Circle these awkward data sets. If you are having trouble reading a number, drop the point onto the axis and make sure you have a clear idea. Use the side of your exam book to be able to draw like a straight line, all that good stuff. If you're a digital tester, you have an average about 1.3 minutes per question. What that means, because you have to make sure you have enough time, right? And there's no way to go back. So use all of your time, okay? So I've always told students that if they don't know the answer, to move on, they can come back. You can't come back. So you need to have some way to tell yourself, okay, I'm moving on at this point. And so like, I guess I'm hoping there'll be like a clock or some way that you can see the time. You need to keep a watch of that time and don't get stuck. Maybe spend about a minute or so when you're looking at your like single question. So it's just a single question. There's no like long prompts or anything. Maybe spend about a minute on that. And then whenever you have the long prompts, you're gonna have to read that and maybe go back to it a couple of times. You could stretch out your set questions a little longer to allow you to accommodate for the going back and forth for the prompt. Also, digital test takers, you could probably ask your teacher for a bubble sheet, or you could print out a bubble sheet. So one of the things that I find is helpful whenever you're going through multiple choice is you can cross out your wrong answers, the ones you know are wrong. You can't manipulate your test. And there's nothing that I've seen where you can actually manipulate the online platform. And so because of that, we find that you can't X things out. So if you have a piece of paper in front of you that already has A through D on it, you can make an X through the ones that you've already eliminated out. So it can kind of help you to jog your memory. Okay, well, I know it's not A and B. Okay, it's going to be C or D. And then like you can actually like manipulate some type of paper. And so with your free responses, the timing tends to be about 25 minutes for your long ones and 10 minutes for your short ones. If you're doing the paper test, I recommend that you do 20 minutes for your long and 8 for your short. This will allow you to have time to go back and read through and add anything. If you're doing the online exam, you better have a 25-minute and a 10-minute. Don't even play games. Use the full amount of time because you cannot go back to it. So go ahead and anything you can look in the diagram, anything you can do, go ahead and do that. You need to have checkboxes. So as you saw with the first response we looked at before, there was 3 different things that we had to do. We had to calculate this number. We had to calculate that number. Then we had to, give this information. Well, make yourself a checkbox to make sure you've actually completed everything, okay? Answer in the order of knowledge. What do you know? Do you know C? And then you could do B? Answer in the order that you know it. You do not have to do A, B, C, D, E order. Just answer in the order that you do know these questions. And then when you're writing your free responses, read your question and read the question. And did I say read the question? I find that as students are doing the free responses, they tend to forget what the question was, and they're giving great biology. They're giving so much information, but they're not answering the question. Label your responses. So especially if you're not going in order and you're just doing D first, then A, label it so that the readers can follow easily. It also ensures that you get the points where you think you're getting the points. So write your knowledge order, as I said before. Beware of a contradiction. So if you tell me that the sky is blue, and then the next sentence you say, well, because the sky is green, you just told me it was blue. Now you told me it's green. No points. So you cannot contradict yourself. You cannot give me two statements. Even if one's right and one's wrong, you can't contradict yourself, because that's just you throwing information out, hoping that the spaghetti will stick on the wall, and that's just not working. Use diagrams and then define your terms. I find that when you don't know the answer, sometimes by defining a term, you can kind of help yourself kind of keep going, and you end up giving yourself the answer. Cross out. And then also, if you're doing the paper test, it doesn't matter if you're using pen, pencil, crayon, or marker. It really doesn't matter. So especially on your graphs, you want to use your pencil to make your initial graph, so that you can if you make a mistake, you can erase it easily. It's not like the pen that you have like issues with. And if there's time, you can go back and do it in pen, but otherwise just leave that thing in pencil. So that was my quick little 5-minute hack at the end, since we skipped photosynthesis for the time being. John?
Yeah.
Is that 5 minutes?
Yeah, this is so great. So thank you. That was amazing. Heroic, flying through all of this. Everyone, thank you so much for coming. We've got a lot of love in the chat for everything you've done today, Tiffany. We have posted this in our Discord server at Marco Learning. I'll post the link to the Discord here. Tiffany and I are both in there. And in fact, let me copy that. And I'll post this in the chat for all of you to join. I encourage you, again, to follow us on, I'm gonna go ahead and, where's
my
this?
screen?
Yep. So I'm gonna post this in the chat here on YouTube. Definitely remember, we are going live, and there are tons of live and reviews and resources that are available. I also, Tiffany, I shared your Linktree page right here in the chat as well. And Ria says, thank you so much for this, Mrs. Jones. It was really helpful.
Of course.
So,
actually, that may have been my student, but I don't think it is.
yeah. So the Discord link is there. I encourage you guys to join us. Thank you for coming. We're going to be posting all kinds of great updates for you in the coming weeks and months. Good luck, and be a bio penguin.
Yes, because you are dressed for success. So remember, you're AP Bio Penguins and you're dressed for success. Have a good day, everyone.
Yeah, that's right. See